Hi everyone, my name is Emily Park, and today I'll be presenting a section of a paper, Patient-Specific iPSC-derived endothelial cells provide a long-term phenotypic correction of hemophilia A. This paper is written by Cristina Agassi and her colleagues, and this paper over talks about hemophilia A patient blood is collected, and from there CD34 cells are selected and reprogrammed into iPSCs. Then the gene is corrected into functioning gene and differentiated into endothelial cells and transplanted into a mice and check if the bleeding phenotype is actually corrected. But today I will be only focusing on this section in the red box portion of the paper, which is generating iPSC. Before we dive into the paper, let's talk about what hemophilia A is, which I will refer to as HA throughout this paper. Hemophilia A is X-linked recessive disorder where blood clotting process is slowed down due to deficiency of clotting factor A. As shown in figure here, when you start bleeding and when you have a lack of clotting factor A, you won't be able to clot the wound and the blood, and you will end up keep bleeding, which is definitely not good. And this deficiency of clotting factor A is caused by mutation or deletion of FA gene. And the frequency of HA is about 1 out of 5,000 in males, which is pretty rare, and it's much more rare in females. Now, what is induced pluripotent stem cells? It is derived from adult stem cells, and it was reprogrammed back into an embryonic-like pluripotent state. And from there, you can develop an unlimited source of any type of human cells. And this method is very frequently used in therapeutic purposes, where, like in this paper, you produce endothelial cells that secrete functional F8 to fix hemophilia A. So let's dive into actually what they did to generate iPSCs. They took the patient's blood, or the subject's blood, and they isolated non-mobilized CD34 cells from their peripheral blood. And they reprogrammed the cell with lentiviral vector, which are viral vector that expresses OCT4, SOX2, and KLF4, and these are all reprogramming factors that produces iPSCs. And as shown in figure, these two are the LVs that were used in this method, in this paper. And how does lentiviral vector actually work? What happens is that the virus that contains those viral RNAs that expresses the reprogramming factors are fused into the cell, and its viral RNA gets reverse transcribed into RNA, viral DNA, and DNA goes into the nuclease, integrates itself into the DNA, and transcribed. And eventually, it can be its reprogramming factors can be expressed. These are some of the list of the subjects that were used in this experiment, and we used 5 of the healthy donor blood and 1 heterozygous blood and 4 of the hemophilia HA patient blood. After iPSCs were generated, they wanted to make sure that they were functioning and they can actually differentiate well and have some good pluripotency in them. So they picked out certain cells based on embryonic stem cell-like morphology, which has oval nucleus and irregular cell outlines. And these two are the example of cells they have picked out, and this is the healthy iPSC, and this is the HA iPSC. And they were able to establish about 10 to 15 clones for each donor. And then using those picked out iPSC, they tested it for AP testing, which measures the amount of alkaline phosphate enzyme in the blood, and alkaline phosphate enzyme is pluripotency marker. So if you test positive, that means you have some pluripotency. And they, as you can see in the figure, it was able to test positive, and that is shown by the color. This color means that it is AB positive. Then they also detected endogenous and exogenous factors And they were able to see that iPSC cells they generated were able to express endogenous recurrent factors, and those are shown in the figure E and F. The column 3, 4, and 5 all show bands for all these factors, and also 1 and 2 and 3 columns for HA iPSC. And but also, however, they noticed that the exogenous recurrent factors were silenced, and that is shown in figure G and H. As you can see, there are no bands present. Then they wanted to detect whether stem cells markers were expressed, and to do that, they used immunofluorescence method, which is used to detect presence of antigen or protein. And in that, what happens is that primary antibody recognizes that protein, and then secondary antibody recognizes that the primary antibody, and that secondary antibody contains a fluorescin and is able to fluoresce. So if the protein is present, then you can see the fluorescence. You can detect the fluorescence. And through this method, they were able to see whether all these stem markers were present. And stem markers we looked for were OCT4, SOX2, TRA181, and SSCA4. And as you can see, OCT4 is shown in green. It's shown in both of these, healthy and PA patient. And SOX2 is also green here. You can see in healthy patient and PHA patient. And also SSEA in green and TRA181 in red. It can also be shown in both. Then they also wanted to observe telomeres. When they noticed that telomere length increased between P5 and P2 iPSC, as shown in the figure O, this is the healthy patient. It increased from P5 to P20, and same for HA patient, it increased. And this increased length in telomere demonstrated a reactivation of the telomerase complex. Also, they were able to notice that 3 germ layer markers were expressed. They noticed that iPSC they generated the iPSC generated embryonic bodies, which expressed 3 germ layer markers, which are ectoderm, mesoderm markers, and endoderm markers. And these are all the list of ectoderm and mesoderm and endoderm markers. And all these were tested to see if they were expressed. And as you can see in that EV column, which is the embryonic body, all bands are present. And some of them are a little faded, but they are definitely present in there. And they were able to see that through that, all those from 3 germ layer marker, all those could be differentiated into adipogenic, which is the R section right here, and osteogenic, which is the X section right here, and chondrogenic cells, which is the T section right here. All the iPSCs were able to differentiate into these types of cells. So in conclusion, all those results displayed that peripheral blood CD34 plus cells were successfully reprogrammed into iPSC expressing pluripotent markers with a good differentiation potential, because you can see that they expressed stem cell, they expressed the 3 germ layer marker, they were and endogenic reprogramming factors. They were like it has a very good pluripotency and differential potency. For future directions, after you generate the pluripotent iPSCs, next step is to differentiate the iPSC into functional endothelial cells, and the corrected gene through gene transfer using LV, and injection of the corrected gene into the mouse model of HA, and observe the functional activity of the F8 to see if the bleeding phenotype is actually corrected. And this is my love, friends. Thank you.